Hello everyone. We are once again in the What We Are Reading YouTube channel of the South American Mycorrhizal Research Network. I'm Cesar Marin, and this time around we have Bia Bock. She's a PhD candidate at Northern Arizona University, and recently, along with Jason Huexima, Kitty Guerin, and Nancy Collins Johnson, they published a very nice paper on communications biology entitled Evidence for Common Fungal Networks Among Plants Formed by a Dark Septate Endophyte in Sorghum bicolor. As you may know, common fungal networks and common mycorrhizal networks in general have been a subject of our YouTube channel for a while. We had previous interviews about this topic to Justin Kars and co-authors, and also to Matthias Rillig. And today would be a third interview. So, hi, Bia, thank you for being here, and tell us how this study came about. Is this part of your thesis?
Yeah, so this study is one of the chapters of my dissertation, and it came about because I was talking with one of my advisors once about common mycorrhizal networks, and she said something along the lines of, I wonder why other fungi don't also form them, because, from a conceptual perspective, there's no reason that they shouldn't. And so that kind of kicked off that whole chapter where I went into diving into what we know about common mycorrhizal networks, and then what we know about other fungi and their capacity to form these common networks, which is we don't know much. And so that sort of kicked that all off.
That's great. And yeah, if you can elaborate on that, on telling us why it's important or interesting to study common networks on non-mycorrhizal gills.
Yeah, I mean, I guess I think it makes a lot of sense that we've focused on mycorrhizal fungi for so far in studying these common fungal networks. And so we've been calling them the common mycorrhizal networks because we only know of them in mycorrhizal fungi. And by that I mean these continuous connections of fungi between at least two plants. And it makes sense that we've focused on mycorrhizal fungi because a lot of them are obligate biotrophs. So a lot of these mycorrhizal fungi require their host plant to live, and they pretty much only exist in their symbiosis with the plant. And so I think that kind of, and because these symbioses between mycorrhizal fungi and their host plants are so famous and have such good establishment, it does make sense that we have focused on them. But in my thinking about this, there are a lot of other fungi that also associate with plants that are not considered to be mycorrhizal, and a lot of these are coexist with mycorrhizal fungi and then also exist in plants that are non-mycorrhizal. And so I think it's interesting to think about the capacity for non-mycorrhizal fungi to form these common networks because, in these systems where we know that common mycorrhizal networks are forming, it's also possible that non-mycorrhizal fungi are forming similar networks at the same time in the same place, doing potentially similar or different things. And then also that they exist in places where mycorrhizal networks are not happening. So, for instance, if you think about the Brassicaceae, a lot of those do have, maybe don't have mycorrhizal fungi, but do have endophytic fungi interactions. So that's kind of a spot where you wouldn't expect a common mycorrhizal network to form, but you could expect a different type of common fungal network to form.
And among those other gills, then this study focus on dark septate endophytes. Can you tell us uh, a bit what dark septate endophytes are? Uh, what are their usual functions, or how people usually study them?
Yeah, I think it's interesting when now that people know that I study dark septate endophytes or DSCs, what people come up to me and say about them. And often it's that you've always seen them under the microscope, but you're never really sure why they're there. You don't always count them. And so they are defined, so they're a polyphyletic grouping within Ascomycota, and they're defined by their morphology. So if we think about dark, they have dark pigmentation, often melanin, and they are septated, so they have those regular cross walls in between cells under, in their hyphae, if you look at them under a microscope. And then they are endophytic, which has kind of a tricky definition in that technically the words endo means within, phyte, plant. So technically they are within plant, but people often use the definition of endophyte where they live within a plant without causing visible disease symptoms. And so that's kind of where endophyte generally the definition is a little bit tricky and people maybe have some disagreements there. But DSCs are often thought to be helping plants in more extreme environments. So, for instance, in super arid environments, you might expect more DSCs. In Arctic environments, you might expect more DSCs. And one thing I kind of have, so I think the dogma is that they are these more extreme endophytes, and maybe their melanization has something to do with that and that it's some sort of bioprotectant. But from what I know, I kind of think that they're just everywhere. And I think that's kind of lovely that they're potentially ubiquitous, and just based on my reading and what I've heard. But people do think of them as more extreme.
Hmm.
Does that answer your question?
Yeah, yeah, totally. And I guess that with them happens the same as with maybe the Mucoromycotina, in which we don't have all the molecular tools to to study them, so they are not often as detected. But yeah, it happens to me that when I analyze roots of the trees that I study, they are there. Yeah, definitely.
Yeah, and actually, so one thing, it's interesting that you bring up the Mucormycotina fine root endophytes, the M-frees. Um, so one thing with DSCs, and even in this paper that we're talking about today, I got some pushback from one reviewer who kind of said, "Well, why are DSCs not mycorrhizal? They sound mycorrhizal." And one distinction is that we don't know of a really precise nutrient exchange that occurs between DSCs and host plants. And in addition to that, we don't know of a nutrient exchange structure that DSCs form within plants. So they do form, specific structures, for instance, microsclerotia, but we don't think that that's a nutrient exchange structure. We don't really have evidence for that. Whereas with emphrees, they kind of recently have been established as definitely mycorrhizal and distinct from AMF. And I think Katie Fields' group has done a lot of really awesome research on that front. And they do form arbuscules and arbuscule-like structures. And there is definitive proof for a nutrient exchange between emphrees and plants. So those are kind of considered um, definitely mycorrhizal, but I mean it's confusing because they do have endophyte in their name, right? So I think there definitely could be some more work done in terms of getting everyone on the same page for what exactly an endophyte is. Um, and actually, one thing I think that is also interesting about M3s, considering that they kind of used to maybe be grouped with AMF, is that they seem to be facultative saprotrophs. So you can grow them in culture without their host plant, which is not the same as AMF. And DSC is the same thing. They don't require their host plant. You can just find them in the soil, free living. Um, but a difference between the two is that, Mucormycotina are constrained to that group, whereas DSCs are polyphyletic. They're a bunch of different groups.
That's that's so interesting, and it reminds me the similar interview that I have with Matthias Rillik, where we got to the conclusion that these boundaries between guilds are very kind of artificial. I mean, they are very context dependent, particularly those taxa which are able to be mycorrhizal and saprotrophic at the same time is very difficult to establish a boundary of the gill. Um, Going into the paper, can you tell us first the three main hypotheses that you had and also the experiments to test those hypotheses?
The three main hypotheses of this study were that, so my model organisms in the study were for the non-mycorrhizal fungus, as you said, used a Dark Septate Endophyte, and in my case it was Alternaria alternata, which is pretty common and interestingly considered to be a pathogen sometimes in an agricultural setting. And the plant that I'm using is sorghum bicolor, which is a pretty common crop plant. And so our hypotheses were first that the DSC would form those hyphal connections between the two plants, so that some sort of physical connection would occur between the two plants. Secondarily, that access to the fungus in that sort of network setting would improve biomass of the host plant. And thirdly, that the fungus would facilitate water transfer from plant to plant. So those were the three main hypotheses. And we studied this in a lab setting, just so as a proof of concept, so not saying anything about how much this actually happens in a natural setting. But so we used these boxes where we grew two different plants, and in the middle of that box, separating the two plants was a special barrier. And essentially, this barrier defines our different treatments for the experiment. And a common criticism point against common mycorrhizal network research is that soil diffusion is not accounted for. So it's possible that instead of nutrients going from plant through the mycorrhizal fungus to another plant, it's possible that the nutrients or whatever it is are just going from plant to plant through the soil just through diffusion. And so what we did in our experiment is we had this special barrier in the middle, which we have a nice little graphic of in the paper, where there's a 3 millimeter air gap between two pieces of plastic. So it goes plastic, mesh, air gap, mesh, plastic, and then the plants are on either side of that. So in order for something, whatever it is, the fungus or water, to get from one plant to the other, it has to cross through the mesh, then cross through the air gap, and then it has to cross through more mesh, and then it can get to the other plant. And so essentially what we're trying to do there is control for that whole soil diffusion factor. And so the three main treatments that we report on in the main text are permeable, which is what I just described, where we have the fungus added to a donor plant and then it can cross through that barrier to the receiver plant. We have impermeable, where the fungus is added to the donor plant, but it can't cross through that barrier because we make it impermeable. And then we have an azenic treatment where it's the same setup as the permeable treatment, the first one I described, but there is no fungus ever added to the donor plant. So it's just two plants, no fungus. And so in that case, if you saw water transfer, then you would know that it's somehow not through the fungus, because and which we did not see water transfer in that treatment, which makes sense. So those were our three main treatments, and we had a whole bunch of supplementary ones as well, which I can go into if you want me to. But yeah, so that's our basic experimental design.
Sorry, and for how long the experiment run? How long the plants grew?
It was about I think 5 weeks total, so pretty small. These barrier or these boxes that we were growing them in are not huge, so they were kind of restrained in terms of space. So we did not grow them for a very long time.
And do you think that the air barrier addresses all the criticisms that have been raised with the common fungal networks experiments?
I think that it addresses some of them. I think that there could still be people who, so for instance, one concept that I've kind of thought a bit about, and especially you mentioned Matthias Rillig in relation to his paper where he talks about the definition, I think it's clarifying the definition of common mycorrhizal networks or common fungal networks. Um, so it's always possible that, so in my experiment where you have the plant, the fungus, it crosses over the air gap to the other plant, it is possible that, that one hypha breaks or something like that. And then, so say, for instance, water is released and then reabsorbed through a different hypha of the same genotype of fungus, and then that goes into the plant. And in my case, in this experiment's case, we used this sort of tracer dye. So we likely would have seen some sort of leakage of the dye, and it was pinkish red in pigment, and it was visible to the naked eye. And we did not see that, but I mean, that doesn't mean that it couldn't have happened on a super small level. So I think that there are always spots where if you take the most strict definition of common fungal networks, that there could still be criticism there. But I think in terms of addressing the main complaints or points of criticism about soil diffusion, this experimental design does address that.
Great. So you tested first it was the connectivity, right? Or the ability of two plants to connect via a DCA common network. Then the effects on plant growth, and then the effects or water transport. So what were the results regarding these three things?
The results were that yes, a DSC can form a common network between at least two plants. So it does form that physical connection. And we did a side experiment that shows up in our supplementary where we just did a simple experiment where we, put them on a clear substrate, put the two plants on a clear substrate, and you can, visually see the hyphae connecting the two plants. So, in addition to our main experiment finding that, that seems fairly well established that the connectivity can occur. Secondarily, with the biomass, we did see benefits in biomass from access to the common network. And one thing I think is important with this point is that I think that the benefit comes from having access to the DSC. And I don't necessarily know, so our experiment doesn't necessarily say that being connected to another plant in that common network is providing that biomass benefit. I think that it's more access to having that common fungal network, the fungal part of it. And that is kind of one of the hypothesized benefits of CMNs in that if a sapling starts growing in a forest, then it can tap into a readily available source of inoculum through this common network. And so I think that's what we were seeing in our experiment with these biomass benefits because it was pretty clear, we would see the fungus cross from box or chamber to chamber and immediately so the plant would be super unhappy and small and bad looking until the fungus reaches it and then immediately its growth improves. And you can see that in the results as well, where the receiver plants are always slightly smaller than the donor plant. And I think that's just because of that 2 week gap basically that it took for the fungus to get from donor to receiver. So the donor plants always kind of had a 2 week advantage in growth over the receiver plants. So anyway, to answer your question, yes, we did see biomass benefits. And thirdly, with the water transfer, we did see evidence for water transfer from donor plant to receiver plant through the DSC network. And one thing I'll note with that is that we did not see it in all of the plants. So we saw it in a lot of the plants, and I think that that indicates some sort of context dependency that we are not quite capturing. So we don't know why it happens sometimes, not all the time. But that seems pretty common in mycorrhizal research generally, where there is this sort of context dependency that we don't fully understand. And so we did see some of that in our experiment. Um, and I think that would be interesting for future experiments too.
Well, yes, I mean, here you are just testing that it happens, of course,
Mhm.
controlled conditions, but then other experiments need to address which factors um, increase or decrease this possible connectivity, and then, of course, under field conditions. But here you show that physically can happen, and that there are these three kind of benefits or these two benefits, plant growth and water transport. And it's also clear to me, according to what you just say, that the benefits on plant growth, for example, by being connected and not necessarily by taking something from the other plant, right? If I understood you correctly.
Yes, yeah, definitely. And I think that's clear from the biomass data where, again, I think that the benefit to the plant in terms of biomass is just coming from access to a ready source of fungal inoculum. And I think that would be interesting to test. You know, in my experiment, there are no other microbes than the fungus that I added. In a natural setting, you have all sorts of microbes and and so I think that would be interesting to think about in that context. And as for the water transfer, I mean, we don't know that it's an ecologically significant amount of water transfer. And so that would be another avenue for future research that I think would be really interesting. But yeah, I mean, to answer your question, I don't think that it's we don't know that the benefits are necessarily because you're getting something from a different plant. Especially in terms of biomass. It seems more that it's just connection to the network.
Okay, that's great. Um, so talking about common networks by DCE and by other guilds. Um, and I'm just wrapping up the interview. Do you think that the field and mycorrhizal ecologists and mycorrhizal researchers are ready for these broader definitions of what a common fungal network is? And because the focus has been a lot on the idea that one single ivy can connect a whole forest, right? Maybe uh, or many plants in a forest, but here you are showing that it's possible with other gills, not mycorrhizal gills. And also we mentioned Matthias' work earlier that IFAL connectivity is not always necessary. So how do you think this research will be in the next 10 or 20 years with these broader definitions?
Yeah, I mean, I am definitely in the camp that I support having a broader definition of common mycorrhizal networks or common fungal networks, and then special use cases like what Matthias Rillig suggested with hyphal continuity being a special case of these CFNs or common fungal networks. I think that some people don't agree with that, and so that will definitely be a point of discussion in the field in the next 5, 10 years is can we get everyone on the same page? I mean, it does seem to me like, I'm working in an experimental system in the lab where I can control for a lot of these factors, but once you bring these experiments to the field, if if you can just discount findings by saying, well, I'm not entirely sure that it was that exact same hypha from that exact same phenotype of fungus that's connecting A to B, that is a pretty extreme barrier, which some people would argue is fine. They might say, well, we just can't study it. We just can't say that those effects are from that fungus. But I don't know. I also think that factors like soil diffusion could be interesting in that if materials move through the soil along with the hyphae and it's faster because of the hyphae, then that seems kind of all part of a relevant system that I think is ecologically interesting and potentially significant. So yeah, I think that it is important for people in the next few years to be very specific with which definition of common mycorrhizal network or common fungal network they are using and what they're claiming. Because, if you're not claiming hyphal continuity and someone thinks that you are, then your results are just different in terms of how you interpret them. So just being really clear with your definitions, I think, will be really important. And I kind of suspect we'll all get on the same page in the next 10 years or so and kind of realize that a broader definition of some sort is ecologically important.
Yeah, and I think that at the end, um, these systems are very dynamic. Uh, I'm sure that, like in the real world, in a forest or in a grassland, at some instances, eiffel continuity might be very important, but at some instances, a messy uh, network of ivy that are somehow connected. Still, there is a lot of movement of nutrients, water, ionosphere microbiomes, a lot of things. So I also lean towards that camp and be very specific with the definitions. And with that, Sorry.
Wait, can I say really one quick thing on that? So what you said reminds me of there's some really good experiments out of the University of New Mexico about this like hyphal loop hypothesis, and it's kind of distinct from the common fungal network realm because it includes non-fungal members, but it's basically this idea that in arid ecosystems that biocrusts can connect spatially dispersed plants. And so you have these kind of monsoon events where you get a big pulse of rain and then you can see nutrients flow faster through the biocrust from plant to plant. And I think that's a good example of what you're talking about, where fungi do seem to be important in that system, and actually DOC seem to be important in that system, but it's not a traditional hyphal continuity because in that system especially, it's not even just fungi, it's other sorts of organisms as well. So I think you're totally spot on with what you were saying on that.
I think that I have seen this word that you are mentioning, and yeah, always when I am in arid ecosystems and I see the spatial patterns of certain plants, I always ask the question, well, how is this connected below ground or not connected? And how those movements of materials below ground explain actually the spatial pattern of the same species or different species of plants. So that's very interesting. Thank you for mentioning that. Um, can you do you have a final message or a final thing that you want to say? And also, what is the next step in your research in this research in specific?
Yeah, I think that the next steps seem to be figuring out how to study these things in a field setting. And one thing I've thought a lot about with the common mycorrhizal network research is that now that we know that other guilds of fungi can also form these common networks, it's not enough to know that mycorrhizal fungi are present and observe the effects. You need to also think about the other fungi that are present in natural settings, because I would not be surprised if DOCs were in other non-mycorrhizal fungi were forming sorts of networks alongside mycorrhizal fungi. So I think that's kind of a wide open area in field research. And obviously, it's a little bit difficult with the whole hyphal continuity aspect, but I think that'll be an exciting realm of research coming up. And final messages, I don't know. Yeah, keeping your mind open to different ideas and seeing, it's if the dogma has always been that mycorrhizal fungi are doing this one thing, then why not anything else?
Excellent. Thank you so much, Evie. I really, really enjoy this interview. And thank you everyone for listening.